Biochemistry Part 1. We'll briefly describe models of molecules and different types of chemical bonds. The fundamental materials that are used to construct organisms go right down to what we call the molecular level. Biochemicals are used in the construction of cells, tissues, organs, and organisms. Microbiologists use biochemistry all the time. We have to describe our microorganisms frequently simply by their biochemical construction, or the molecules that they use for food or that they produce as products. Now, in some of the following PowerPoints, we will go over in more detail the four major biochemical groups of biological molecules: the nucleic acids, proteins, lipids, and carbohydrates. But for these few slides, we're simply going to get the introductory information first. There are two ways that biologists frequently describe biological molecules. We can use the molecular formula that simply tells us the number of atoms of different elements that are used in the construction of the molecules, and we can also use little pictures, or what we call the structural formula, that simply illustrate, using pictures, the atoms of the different elements that are used to construct the molecule. The biggest difference, the molecular formula doesn't tell us how the atoms are arranged. The structural formulas, or the structural pictures, are very important to give us an idea of exactly how the different atoms of the different elements arrange themselves spatially, so we can figure out just what a molecule looks like. Here are some examples of other kinds of chemical or structural models. We can have what are called essentially carbon skeleton, or ball and stick, or space filling. There's all types of ways that scientists use drawings or illustrations to describe how a molecule is arranged and how it fills up the space in which it occupies. Molecules that are made largely of the element carbon are said to be organic molecules. Frequently, those organic molecules include not only carbon, but hydrogen, oxygen, and nitrogen. In fact, carbon, hydrogen, oxygen, and nitrogen are the four elements that make up roughly 96% of all organisms on Earth. If molecules are constructed from other types of elements, we might often describe those as inorganic molecules. So organic versus inorganic often depend and determine where we would find these molecules and what kinds of elements are used to make the molecules. The glue that holds elements of different atoms together to form molecules are called chemical bonds. Now, it's not literally glue, but it's ways that different atoms of different elements can arrange themselves to hold together to form molecules. In order to understand how chemical bonds are formed, we need to understand the structure of an atom. An atom is a representative structure of an element that's comprised of an atomic nucleus at the center of every atom, which is constructed of protons, and there might be neutrons. Protons are positively charged, neutrons have no net charge, so they're neutral, and then zipping around or floating at a distance from the nucleus are negatively charged electrons. Now, the number of protons in an atom of an element determine the characteristics of that element. And so we say that the number of protons is actually the atomic number. If we total up the mass of all of the components of an atom of every element, the mass of the protons, the mass of the neutrons, and the almost negligible mass of the electrons, that total mass of all of the components of every atom of every element is called the atomic mass. Now, if we take a look at this diagram, notice that we have an illustration of an atom of sodium. We have the nucleus nicely packed with 11 positively charged protons, 12 neutrons that have no charge, and then we have an equal number of negatively charged electrons essentially circling or floating around at quite a distance from the atomic nucleus of the sodium atom. So we have overall a neutral charge on this atom because we have an equal number of negatively charged electrons that equals the number of positively charged protons. Now, the electrons in the outermost layer or shell, there are different ways that chemists describe where these electrons are, and they really aren't floating around in these little circles. This is just an illustration for convenience. But we do find that there is one level or area at the outermost surface of an atom where we will find some of the electrons distributed. This outermost shell is called the valence shell. And ideally, for most elements, we find that nature wants to put, at least for the elements that we find in organisms, 8 electrons. Now, that's not going to be found in every element. Every element's going to have a slightly different number of valence electrons. And notice here for sodium, in this outermost surface of this atom, we have one lonely little electron in its valence shell. Now, this is going to be important when we want to construct a type of chemical bond using an atom of sodium. Now here we have an example of how different atoms of different elements can essentially help one another by exchanging electrons from their valence shells. Some atoms that have very few electrons in their valence shell, like sodium, with 1, could easily donate an electron to a different element, like chlorine, that has a valence shell of electrons of 7. Now notice, if we want to have these two different atoms react with one another, it can be, in fact, a give and take kind of arrangement. Sodium will donate its outermost single valence electron to chlorine. Chlorine will accept that electron to fill out its valence shell, so it now has 8 electrons, ideal for a valence shell. So we now have sodium has 10 electrons total, and chlorine now has 18. Sodium is missing one of its negatively charged electrons, which means there's a proton in the nucleus that now can express its own positive charge all by itself. With chlorine, we've actually accepted an extra negatively charged electron. So we now have an additional negative charge on the chlorine. So now if we take a look, sodium that is now positively charged and chloride, which is the name we now give to chlorine once it's gained an electron, are now called ions. When an atom gains or loses an electron, we no longer call it an atom, but instead we call it an ion. If it's a positively charged ion, we call it a cation. If it's a negatively charged ion, we call it an anion. Once we have positive and negative charges expressed by ions, those positive and negative charges can be attractive to one another, just like magnets attracting one another. This attraction, because of the gaining and losing of electrons, establishes the formation of what's called an ionic bond, which is the first of 3 different kinds of chemical bonds that we need to know about for this semester. So this is an excellent summary slide. This would be a good one to use to study for the different characteristics of ions, the formation of ionic bonds, and the definition of cation and anion. Remember, we've taken 2 different atoms of different elements. They had different valence electrons in terms of numbers. When one element donated or lost its electron to a different atom that gained it, we formed cation and anion, which would then attract one another to form ionic bonds. Sodium and chloride ions, when they have these kinds of ionic links between one another, ionic bonds, form essentially table salt, which is what sodium chloride is. The second type of chemical bond that we need to know about are the hydrogen bonds. Hydrogen bonds are weak attractions between the hydrogen of one molecule to the oxygen or nitrogen of a different molecule. Typically, a good example of the formation of hydrogen bonds is depicted in water. Water is essentially, in its liquid form, held together by what we call hydrogen bonds. The weak positive attractions and negative attractions between the different water molecules. If you take a look at some of the little illustrations of a single molecule of water, notice that we have oxygen and 2 hydrogens. Now, the electrons for the hydrogen atoms are actually going to be shared or form what we call covalent bonds, which is the third type of bond we'll talk about in a few more slides, with the oxygen atom. And in fact, the oxygen atom is so much more massive, or what we call electronegative, than the hydrogen atoms. The electrons of the hydrogen atoms spend more time circling the oxygen nucleus. That results in a distribution in a slightly negative charge around the oxygen atoms and a slightly positive charge around the hydrogen atoms. We depict this slight positive and slight negative charge with the Greek symbol delta. Now, in modern terms, the lowercase version of the Greek symbol is more commonly used, though in older textbooks you can find either upper or lowercase of the Greek symbol delta with a positive or negative sign next to it being used to depict this partial positive or negative charge. These weak attractions of one molecule to another are very important in all types of interactions in biological systems. In fact, it's the hydrogen bonds that are often very important in holding molecules together just loosely enough to allow biological processes to occur. This slide simply illustrates some of the properties of water in pictures based on the charge distribution, or the fact that part of the molecule is positively charged weakly and the other part is weakly negatively charged. With this different charge distribution over the surface of the same molecule, we say these molecules are polar. And because of the polar nature of water, it allows water molecules to interact with one another. The third type of chemical bond we need to know about are the covalent bonds. In covalent bonds, valence electrons are shared. We don't lose and gain like we did with ionic bonds, and we don't have partial positive and negative charges like we did with hydrogen bonds. Instead, covalent bonds are the strongest chemical bonds that we will discuss. They hold things together. So we have different numbers of electrons that can be shared between different atoms of different elements. In the illustrations on this slide, it's depicting both a single covalent bond between two hydrogen atoms and a double covalent bond between two oxygen atoms. But covalent bonds can occur between different atoms of different elements too. Carbon to hydrogen, oxygen to nitrogen. So these are just examples. Now, when chemists draw or write out a chemical structure in a molecular formula or a chemical structure, they might simply use letters and lines. The letters, like the H, is the abbreviation for the element hydrogen. The O would be the abbreviation for the element oxygen. And then we're simply going to draw either 1, 2, or maybe even 3 straight lines between the abbreviations for each element that's in part of the molecule that we are describing that's being held together by covalent bonds. If we draw a single dash between the letters for the different elements, that's simply a scientist's abbreviation for a single covalent bond. If we draw 2 lines, like an equal sign, between the 2 abbreviations for the different elements, that's describing, or a chemist's abbreviation for, a double covalent bond. Now, we can find in nature triple covalent bonds can occur, such as nitrogen gas that's in our atmosphere. 2 nitrogen atoms are held together by a triple covalent bond to form nitrogen gas. But those are inorganic examples, and in fact, in organisms, we only find single and double covalent bond examples. Here we have an example of a single covalent bond between 2 sulfur atoms. So as we had with the previous slide, this is simply the sharing of electrons between 2 atoms. These 2 atoms could be found on parts of structures that are quite distant from one another until we start to fold or bend the parent structure. We'll come back to this as an example, but when we have 2 sulfur atoms covalently bound together, we call that a disulfide bond. And the disulfide bond is going to be important when we talk about certain kinds of proteins.